Immunity developed by the attack of pathogens, right?
Okay.
Until unless the pathogen won't attack, the antibodies production will not take place, right? Immunity develop. So
Yes.
basically, the development of immunity. First, I'll just give you an idea. Then we'll go through your textbook, right? So development of immunity. So first type of the immunity, that is uh, passive immunity, right? Passive. So passive immunity is not developed by the we get it by from someone else's body, right? So first passive immunity, whenever we take the birth, first time when we take the birth from our mother's body, right? When we take birth, we get after birth, after birth from mother. So what happened when birth of a child takes place? So first, some sort of immunity comes to his or her body, its body, from its mother, right? Then it is suggested that for first few days, the milk feeding or breastfeeding is really very important, because first the milk which is produced of yellow color from our mother after the birth, so that milk is called colostrum, that is of light yellow colored, and that colostrum have lots of immunity, right? So that colostrum gives immunity to a newborn child. So that newborn child have not developed immunity on its own body, it have taken immunity from outer source, and which is the outer source, that is the mother, right? So that kind of immunity which we take from outer source, that is called passive immunity. Did you got that? That immunity did not develop in your body.
Yes.
Okay. One more thing. You must have seen, Affaf, that when like 2 years back when COVID was in its peak, so there was a technology or medical technology. By the way, that was a flop technology. That was not a successful technology. Plasma transfer. Have you listened about that? Plasma therapy?
No.
No,
No.
plasma therapy? No.
No.
Actually, the plasma therapy was a type of therapy where what we used to do, suppose that a person, suppose that someone else, suppose that I got the COVID, right? So, but now my body is not responding that quickly. So, what will happen? Those person who have healed up or cured from the corona.
Oh, yes. I know about this.
Okay. So their blood used to be taken and their plasma used to be extracted, and their plasma will be inserted in my body, right? So I have taken immunity from, means antibodies from
That's dumb.
another's body, right? So that is a
Yeah.
passive immunity, because that immunity have not developed in my body. Did you got that thing?
Yes.
So we can say the passive immunity, the example of passive immunity, which is like plasma therapy, like vaccine, right? That is the all passive immunity. Now, what is the X second? What is the plasma therapy, or we can say sometime we give platelets from our body, others body, someone else body, right? Like plasma therapy, and we can say one more thing like uh, platelet transfer, platelet transfer. That is also right. So these are the passive immunity. Second, so they are taken from outside of body. Second kind of the immunity is called active immunity. Active immunity, and I have told you active immunity develops in its own body or person's own body. So develop in the body of person itself, right? So in active immunity, first what happened? Suppose that a bacteria or a viral infection it came, right? So pathogen infection, what will happen? So first thing, pathogen infection. When the pathogen infection like virus, bacteria, they attack in our body, what will happen? The WBC, B cells, T cells, lymphocyte, and all that, they will start producing the antibodies. That antibodies will keep on spreading in our body. So first thing, why pathogen attack. Second thing, we do not wait for the pathogen. Suppose that I did not got the COVID. What will happen? I have taken the vaccine. Vaccination. What is vaccination? Triggering the. We have already discussed. Vaccination is what is triggering the immune system. Yes, Affaf, right? So,
Yes.
then the immunity will develop in our body. So vaccination, right? So that will be artificial means vaccination part. So this is the type of, that's how the immunity develops in our body. And you must have seen one more thing, that our body is capable of producing like 75,000 type of the antibodies, right? So those child which are rag pickers, which lives in unhygienic conditions, they hardly get ill or sick. And those kids which are pampered, which kept in the house with a very hygienic, you must have seen that thing practically. You
Yes.
you
Yes.
pamper your small kid at your house and you keep it clean and hygienic, then there is a more chances of getting ill of that boy, because immunity have not that much developed in his or her body, because the exposure to the pathogen is least, right? And those poor people who are living in unhygienic condition, they are the rag picker, they are the very poor kind of the people, they are living they are surrounded by the lots of infection. They have more number of antibodies in their body, so their immune system is more responsive and their immune system is stronger than those kids which are living in hygienic condition. It doesn't mean that start living like unhygienic, right? Okay, so got it, Afaf? Afaf, hello.
Hello.
Yeah,
Sorry, sir. Issues with the internet.
no issue. So did you got all the concept and noted down this one, flowchart?
Um, well, I only noted down till vaccination.
Okay, fine.
Sir,
Now,
shouldn't vaccination come under passive immunity?
no, that's active immunity, because the immunity develop in our own body. It is developed by we do not take it. Vaccination is, what is the vaccine? Vaccine is not antibodies. Vaccine are weak or dead pathogens, right?
Okay.
So
So, well, you continue.
when you incorporate that vaccine, means you are introducing weak or dead pathogen to someone's body, then what will happen? That vaccine or pathogen, that pathogen will trigger immune system, and your own your own WBC will start developing antibodies. So the development of antibody taken place at your own body. So that will be type of passive active immunity. Got it?
Okay, so passive immunity is passing of the antibodies
Exactly.
into the
Passing
body.
of the antibodies
External
from
source.
outer source to someone else.
Okay.
Got it? Clear?
Okay. Yes.
Okay, fine. So let's go through with your textbook. Okay. So different type of, as you have learned in section 6b2, when your body comes into contact with a foreign antigen, your immune system activated, it makes antibody needed against the antigen on the pathogen, and the pathogen is destroyed. This is known as the natural active immunity, because your body actively makes the antibodies, right? When mammals such as camel, goat, human, human is pregnant, preformed antibodies passed from mother to the developing fetus through placenta. After the birth, baby receive the additional protection from the antibodies present in their mother milk, right? That colostrum, right? The milk is made up of first few days after giving the birth. It is written over here, colostrum. It particularly rich in antibodies from the mother that gives baby temporary immunity and many different disease until its own immune system start working. This is known as natural passive immunity. Got it? Active immunity, passive immunity.
Yes.
Okay.
Yes.
So these are the natural, right? And vaccination is artificial passive active immunity.
Yeah.
Artificial, right?
Yes.
It's it is passive because your body does not make the antibodies. That's what we discussed. It tend to be quite short-lived because antibodies are not replaced, but very important for the survival of newborn mammals. Right now, inducing immunity, right? That inducing immunity means inducing, that is triggering immunity, means artificial immunity development. People spend a great deal of the money on medicines to treat infection. Another way of controlling the infection disease to prevent them, helping by using the immunization. Immunization is the process of protecting people from infection by giving them passive or active artificial immunity. You develop immunity to a disease causing pathogen by exposing your immune system to a specific antigen in the safe way. Does not put you in the risk of developing disease. It means the virus or bacteria should be very weak. It should not be. You develop immunity to a disease-causing pathogen by exposing your immune system to a specific antigen safe way and does not put you risk of developing disease. By exposing your body to safe from the pathogen, your body produces antibodies and memory cells that are ready to protect you exposed live disease. Vaccination is procedure by which immunize people to produce immunity. The aim of immunization is to protect individual against disease that might kill or harm them. It also have a wider role in society. Immunization can eradicate, elimination, eradicate and eliminate the controlled disease that causes the large number of the death, disabilities, illness with a population and therefore create a serious problem for the structure of society. Immunization like polio eradication, artificial passive immunity. It is given to antibodies from the one individual are extracted and injected into another individual. Plasma therapy, right? Got it? The antibodies may be from a person who is already immune to disease or from completely different species like horses whose antibodies are effective. That does not confer a long-term immunity because antibodies are generally broken down and not replaced. However, it is extremely valuable if someone has been exposed to rapidly acting antigens such as tetanus, right? Jaw lock. So, a result from the toxin and produced by the bacterium Clostridium tetani. So, in tetanus, we know that there are bacteria called Clostridium tetani produce a toxin, and that causes the lock of the jaw and the person die. The toxin affect the striated muscles and causing them to go into spasm like tetanus. This makes swallowing and breathing impossible to cause the death. If someone may have been exposed to tetanus, for example, from the bad cut while tending the crops or working with the horses, they are injured with antibodies against the tetanus. The prevent this prevent the development of the disease and death, but will give long-term immunity, but will not give long-term immunity. Got it? Artificial passive immunity. Now, what is the artificial active immunity? Vaccination, that's what we studied. It is the basis of most immunization program. Here, the small amount of the safe form of the pathogen known as the vaccine are used to produce immunity in the person. The pathogen is made non-effective without reducing the ability to act an antigen. This can be done in a number of ways. If it is a toxin that cause symptom or detoxified from with one or more chemical group change will be injected. Someone inactive virus or dead bacteria are used to vaccinize. Sometimes attenuated pathogen, that is viable but modified, they cannot produce disease. Attenuated means they are viable, means they are living, but they do not produce the disease, are used. Modern vaccines are increasingly produced using the fragments of outer coats, whereas bacteria, even DNA segment can be used. With the artificial active immunity, your immune system will produce antibodies against the antigen, and appropriate memory cells will be formed without you becoming ill. If you subsequently come into the contact with active antigen, it will destroy rapidly without you experiencing the symptoms of the disease it causes. So the same thing, this is the same diagram. Eradication of disease, like polio we have eradicated, right? Leprosy eradicated from the world, WHO says. For centuries, smallpox was greatly feared. The first disease was smallpox, which was eradicated from this world. For the centuries, smallpox was greatly feared. It kills millions of the people and disfigured many more around in the world. Now it disappeared. Smallpox is the only disease that has been completely eradicated by the vaccination program. However, polio is close to be eradicated and as well. Smallpox is no longer found either in people, animals, or anywhere in the environment. The virus only exists and isolated into top security labs. Smallpox were very recognizable and non-human host, a long incubation period, and visible scar as evident of immunization. These features all help us to make eradication possible. You must have seen these scars. Have you seen that? It must not be with you. It must be with your father, your mother, arms of your elders one, who have got the this smallpox vaccine. Have you seen that, Affaf? In the hands of your mother or father, these kind of the scars.
Not on the hands.
Not in the hands. That may be in legs or
No, on the upper arm.
yeah, upper arm. Yeah, that upper arm. This part, 2 or 3.
Yeah.
So, yeah. So these were the scars which comes due to the these smallpox vaccine. Elimination and control of disease. There are many. There are many diseases for which the pathogens survive in soil, water, animal host. Such disease are such diseases. There is always a very large survivor of potential infection. So eradication is not realistically possible. The aim of immunization is therefore elimination and control. Means those diseases which are found in some like other host or some other sources. So that is not practically possible. You can eliminate from the humans, but that will survive in the source where it is found. Elimination is the situation in which a disease disappears in defined area, but pathogen remains in animal, the environment, or in the mid-infection, which cannot recognize. In this case, immunization must be continued even when no clinical cases are observed. For example, vaccination program against polio has not eradicated the disease. Even in countries where the number of the people who are vaccinated is high. A live vaccine is used, so very occasionally in infant or vaccinated carrier develop the disease. In spite of this, polio remains endemic only in Afghanistan, Nigeria, and Pakistan. Scientists or the doctors are working hard to introduce the universal vaccination program in these countries. For example, the UAE is working closely with the WHO to eradicate the polio, especially focusing on the Pakistan and Afghanistan. The Emirates polio campaign has delivered in the total over 86 million doses of the polio vaccine areas where they are desperately needed. Uh, if if threat of the polio is removed, it will be because using the global community is working together like this. A disease that controlled still occurs but not frequently enough with significant health problem for some of the serious infection disease such as malaria, or the rapid evolution of the pathogen makes it very difficult to develop the vaccine because the surface antigens are always changing. So we have already discussed that those pathogens which are changing their constitution of the DNA or their structure, not possible to make the vaccine against them. Like the COVID, it was really very difficult. We made the COVID vaccine. That is a different scene, but it was really very difficult on initial days. Polio, whooping cough, TB, measles. Thousands of people were killed and disabled by disease every year before the arrival of the vaccine and better living conditions for all. In other cases, only vulnerable people vaccinated as general rule, with the mass vaccination to provide a herd immunity only introduced when there is a serious outbreak, right? So,
Sir, I guess it's related to the other paragraph.
sorry. Oh, yeah, I missed that. This one.
Yeah.
Herd immunity, right? The disease to be eradicated, eliminated, or controlled. Herd immunity is important. Herd immunity occurs when a significant proportion of population is vaccinated against the disease, right? This makes it very difficult for disease to continue affecting a population because a pathogen cannot survive without the host to be infected, and very few people are vulnerable to it. Herd immunity can effectively stop the spread of disease through the community. It is important that everyone who can be vaccinated is vaccinated. This protect the individual against the disease. Like very interesting thing, in our country, like in India, government of India have given the free of cost. Like we were the first one to develop the vaccine after the, not after, parallel to the United States. India was the first country to develop its own vaccine, and that was effective vaccine. And after that, this was verified by the WHO, Russian government, American government, and all that. Then what happened? The government of India have introduced 3 doses of vaccine, like first initial doses, then second doses, then the super dose. The super doses now this is continue. It is all free of cost, but still there are many people who have not taken the vaccine in spite of government have given free of cost, pushing it hardly. Still, many people do not take it. Even the government have made compulsory that if you are traveling with the planes, if you are traveling with some sensitive area, then you must have the that certificates of the vaccination, right? Uh, but still many people do not take it due to the various reasons, right? Some of them used to think that it will make them important. Some of them used to think like different things, right? So there were some due to the like illiteracy, right? Due to the illiteracy or due to the some hardcore thinkings, so many of the people, so like in these cases, these are the also hurdle to eradicate a particular disease, right? So government pushes it. The same thing is happening over here. Look, so hard community means like we have eradicated. We have given the doses to particular population. So there will not be any carrier. So the disease will be eradicated in that particular population. So these kind of the immunity is called hard immunity. Herd immunity can effectively stop the spread of disease through the community. It is important that everyone who can be vaccinated is vaccinated. This protect the individual against the disease and also protect people who cannot be or have been vaccinated. These include very young babies, very old people, people who compromised immune system, and the people who very ill with other diseases. Because everyone else vaccinated, the people who cannot be vaccinated will be probably never encountered the pathogen. The percentage of population that need to be vaccinated to give herd immunity varies from the disease to another disease. It depends on the factor that how the disease is spread and how the infection is to give herd immunity against the whooping cough, pertussis. Whooping cough is called pertussis, right? So it is also called the black cough, right? 92 to 94% of the population need to be vaccinated for measles. It is 83 to 94% for polio. 80 to 86% of the population need to be vaccinated against the disease to protect everyone. Some disease are also serious that may that many countries, including the Republic of Maldives, UAE, UK, all young children are affected. All young children are offered the vaccine against them. This gives hard community to whole population. These disease include the diphtheria, tetanus, and polio. So there is a vaccine that is given free of cost to every child in this country. That is called DPT. DPT, diphtheria, pertussis, and tetanus. So that is a combination of single vaccine. The single vaccine, when a young one, when a child take birth in our country, government gives it free of cost. In 1 rupee, you have to spend 1 rupee. Government will give free of cost that vaccine, and that vaccine is combination of three. So when a young baby become of 1 month, you can go to the government hospital and you can take that vaccine. That is called DPT. Right now, whooping cough, TB, and measles. That is the second vaccine. Thousands of people were killed and disabled by this disease every year before the arrival of the vaccine and better living conditions for all. In other cases, only vulnerable people are vaccinated as a general rule. Means if we talk about the rest of the disease, so only those people are treated who have got infected, right? Okay. Vaccination, artificial active, attenuated pathogens, herd community, antibiotic, and treating bacterial disease. What are the antibiotics? Tell me first.
They provide immunity and kill the bacteria present in the body.
What was the what are the antibiotics, and how does it came in the scene? Like, which was the first antibiotics?
I don't know which one was the first biotic antibiotic.
Let me tell you. Drugs again, microbiome and bacteriocellus reticulocyte. Okay, so okay, let me tell you what is the antibiotic. Look, there was a scientist who have was working on the microorganism, and he saw that there is a fungus. So fungus you must have studied or not? Fungus.
Yes.
Okay. So these are the rhizoids of fungus. Tell me what is the name of this fungus? Can you make the diagram?
I don't know.
You can recognize it by this diagram only. These are the conidia. Conidia, conidiospores.
No, actually, we haven't studied types of fungus, but
Okay, okay.
only
So,
just introduction of fungus.
okay, I got it. The name of this fungus is Penicillium notatum. Penicillium notatum. Right? And Penicillium notatum, the scientists have seen that around the Penicillium notatum, no bacteria can survive. Right? If the bacteria will come over here, it will die. Right? Bacteria will die. Right? Die. Why the bacteria die? Actually, this fungus secrete a special kind of the substance, right? From its rhizoid. It is secreting a special kind of the substance, a special kind of the chemical from its body, and that chemical, this chemical, it kills bacteria. So this is the antibacterial chemical secreted by fungus. And the antibacterial chemical that secreted from this fungus, the name of we call these kind of the substance what? Antibodies. Sorry. Uh, uh, what is it? Mm,
Antigen?
no, no, no. Oh, it's just like antibiotic.
I only know two antis.
Antibiotic. 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 So all those kind of the chemical which kills the bacteria are known as antibiotic. Right? Got it?
Yes.
When I was teaching in a few classes back, you must be aware. I have told you there is a that a plant which is Selaginella, right? Selaginella, and that plant which produces some chemical, and in that where it is found in that water do not have any bacteria, Selaginella, right? It used to be in Second World War. It has been used in place of the cotton. Remember?
Yes. It was a long time ago.
Right, right. So that was also that plant also produces like people people plant, you know, because
Yes.
religiosa people plant, that is also very pious. That is also considered next to the god because you know that plant produces some chemical from its root that the antibiotics, and because of that antibiotic chemical, bacteria do not develop around that plant. Right, that's why we consider it is important. So can you note it down?
Good. Okay, I'll note it down. Mm. Okay, so the chemical that's secreted by the fungus is called antibiotic.
Any chemical that work against bacteria is called antibiotic.
Okay.
Who discovered the antibiotic?
I don't know.
Alexander Fleming, if I'm not wrong.
Alexander who?
Fleming. Alexander Fleming was the person. Let me check it. I think the Alexander Fleming should be the person. I was trying to recall it actually. Just my memory is not working right now because I'm exhausted.
I'm always exhausted.
Actually, just before taking 2 classes, I just drive 300 kilometers, so my memory is not working. Alexander. Yes, I'm right. Alexander Fleming was the person who discovered the vaccine. This one, penicillin. So Alexander Fleming was that person. It's working. I had doubt on my body. Alexander Fleming was that person who saw that there is a fungus named Penicillium notatum, and no bacteria can grow around this fungus. So he discovered how does it take in place. Then he came to know that this fungus is secreting a type of the chemical, and he given the name of this chemical penicillin, right? And this penicillin was a type of antibiotic. So the chemical which is secreted by this fungus is known as penicillin. Still, it is used, penicillin. So penicillin was the first antibiotic which were discovered by Alexander Fleming, and still it is used. It can kill wide range of the bacteria. So very effective. Still, we use the penicillin. So penicillin was the first antibiotic discovered. Got it? Got it?
Yes. Done, sir.
Done. Okay.
Yes.
So this was the Alexander Fleming who discovered the antibiotic. What are the antibiotic? Which work against bacteria. Which was the first antibiotic? Penicillin was the first antibiotic. Who discovered it? Alexander Fleming. Where does it come? Penicillium notatum. What is Penicillium notatum? It's a type of a fungus. Clear?
Okay.
Okay. Fine. Drugs again, microorganisms. Big advancement in the treatment of bacterial infection was discovered and manufactured of the first antibiotic in the first of 20th century. Antibiotics are medicines that either destroy the microorganism or prevent them reproducing. Right? So what are the antibiotics? Destroy microorganism or prevent reproducing. They are used to treat patient suffering from the bacterial infections, right? And have had dramatic impact on the human survival of the life exceptionally. In 21st century, it is hard for us to imagine how many people died of communicable disease just over 100 years ago. In 1900, 53% of the death in the USA were from the infection by disease. By year 2010, infectious disease caused only 3% of the US death. This in spite of the emergence of the HIV/AIDS and linked rise tuberculosis. The reason for this, this partially the result of immunization, and partially because of the antibiotic meant which we cured the disease which prevent to kill us, right? Which previously killed us.
Sir, antibiotics kill every kind of microorganism, both good and bad.
Most, yes, yeah. That's what we discussed. Like they kill all good and bad. So what happened when we take the antibiotic, high dose antibiotic, we just start lose motion and all that because good bacteria, good flora also die. All modern antimicrobial drugs work against microorganisms by the principle of selective toxicity. Now, what is the principle of selective toxicity? They interfere with the metabolism or function of the pathogen, right? Particular pathogen, right? They interfere with the metabolism or function of pathogen with minimal damage to the cells of the human host, right? They damage minimum, right? The most commonly used, best known antimicrobial and antibiotic, they can also be used but limited degree in the treatment of the some fungal infection. The first antibiotic to be widely successfully used in the treatment of the bacterial disease was penicillin. Penicillin. Penicillin does not work against all pathogens. So scientists searched more antibiotic and continue to searching today. Many of the antibiotics. Still, we are working on that. Many of the antibiotics are being discovered, have been discovered, but globally the penicillin and many antibiotics derived from the still are it is still commonly used to combat the bacterial infection. Antibiotic action. Antibiotics are effective because they disrupt the biochemistry of the bacterial cells. Different class of the antibacterial interrupt the different processes. The main class of the antibiotic drugs and the way affect the microorganism are summarized in table. Sulfonamides, antibiotic that interrupt the metabolic pathway and block the nucleic acid synthesis, means DNA replication, right? And synthesis of the vital nutrients such as folic acid in bacteria.
Sir, should I learn this table?
Yes, because there are very few, known, and they are very popular. So you must be aware with the sulfomides, tetracycline, very right, beta lactamine, right? So take the actually some take like try to read out. Remember these, they are 4 or 5 only, right? So just this is the way that DNA. So what exactly they do, and their ultimate the function is stopping the bacteria, right? When you take an antibiotic, it may have one or two different effect. It may be bacteriostatic, which means that antibiotic used completely inhibit the growth of the microbe. This level of the treatment is usually sufficient for the majority of the everyday infection. The antibiotic combined with the actions of our immune system will ensure that the pathogen will completely destroy. Tetracycline is an example, right? So that is called bacteriostatic, which will completely finish the bacteria. Example, tetracycline is example of bacteriostatic antibiotic. It is used to treat acne, urinary tract infection, respiratory tract infection, and chlamydia. Sometimes a drug or given dose of drug bactericidal means that will destroy almost all pathogen present, right? This type of the treatment is often used in the severe and dangerous infection. It is also used to treating the infection which the patient immune system is suppressed. In this case, for example, in the transplant of the patient, when our organ is transplanted, who take the immunosuppressant drugs protect transplant organ and certain disease such as TB and HIV. Penicillin drugs related to the origin penicillin are example of bactericidal antibiotic which is used to treat the skin infection, chest infection, and urinary tract infection. In fact, the term bacteriostatic and bactericidal are misleading. How they work dependent on the dose given. Bacteriostatic antibiotic often kill a lot of bacteria and will usually kill them all to at high enough doses. Bactericidal antibiotic do not kill all of the bacteria. They kill 99% within the given time frame. Lower doses of bactericidal antibiotic are often bacteriostatic. So these definitions are flexible. Means sometimes bactericidal also become bacteriostatic, right? Broad spectrum antibiotic destroy wide range of harmful bacteria. Like penicillin have broad spectrum. Broad spectrum antibiotic, broad spectrum and single vaccine, it kills number of the macroorganisms, number of the bacteria. So we say it have the broad spectrum. Narrow spectrum, when antibiotic kills a very few bacteria or very few type of the bacteria, we call it narrow spectrum antibiotic. Broad spectrum antibiotic destroy the wide range of harmful bacteria, pathogen, and neutral good bacteria alike, right? It is mentioned over here. A narrow spectrum antibiotic target one or two specific pathogens. Factors in effectiveness or antimicrobial microbial drug include the concentration of the drug in area of the body infected. This will affect by how easily the drug can reach to the tissue and how quickly it excreted. The local pH, whether either pathogen or host tissue destroy the antibiotic, the susceptibility of the pathogen to a particular antibiotic used. If the standard dose of drug with a doctor normally prescribes successfully destroy the pathogen and cure a disease cure the disease, the pathogen is said to be sensitive to the antibiotic. If disease is cured only by using a dose of antibiotic is much higher than the standard dose, then the pathogen regarded as moderately sensitive to antibiotic. However, there are increasing number of the cases where a particular microorganism is not affected by all antibiotics. Sometimes even that many have been effective in that past. In these cases, microorganism has become the resistant to antibiotic. This microorganism is called antibiotic resistant. Did you got it or not? And we have, when I was teaching you the structure of the bacteria, I told you that the plasmid at that part which develop the resistance for antibiotic, and every time it become more and more resistant to for particular antibiotic. Is it clear, Affaf? Is that clear?
Yes.
Okay. Practical string. Is that practical? Did you have performed any practical?
Not in school. I've read about it.
Should we go through it? Practical or not?
Yes.
Okay. So practical skills, read it out. You can investigate the effect of different antibiotic on bacteria using standard microbial technique which you discovered in. You can inoculate agar plate. We have already studied that how to develop the bacteria. Yes, remember?
Yeah, I remember.
You inoculate agar plate with known bacterial culture. You place a filter paper disc containing different antibiotic of different concentration on the same antibiotic on agar. You cover and do not seal the plate, so oxygen is not excluded. You go a control culture of microorganism with known sensitive to antibiotic at the same time undergo the same condition. You measure the effectiveness of the drug by the level of inhibition of bacterial growth. Different culture, give the antibiotic and check it, like either it is controlling the bacteria or not. So
Sir, in when I have to write about these investigations or experiments, I also have to write down safety precautions and what
yeah,
equipment is
yes,
needed.
you have yeah.
So what would be the safety precaution here
Safety
and what
precautions that
equipment?
you must use the mask, you must use the and the experiment should be like you must always while disposing the microorganism, we have already understood that while disposing the microorganism we must attenuate all the equipments which you used and dispose it properly. so that they do not go for another, they do not spread the further infection. And while performing the experimental lab should be separate. It should not be domestic lab and all that. Those are the precautions. That's it. Mean you must not be, the precautions are you must not be infected, get infected from that experiment, and after disposing, someone else might not be, or society might not be infected, right? That is the only
Okay.
precaution, right? And next
What about equipment?
equipment, these should be attenuated with the help of spirit and all that, right? And one more thing, that while performing the experiment, that culture should not be infected from any virus
No,
and all
sir.
that.
What I mean is like what things are needed. Like for example, conical flask and other things.
No, we need not to mention all that. Just we need the petri dish and the cultural plate. That's it for the bacteria.
Okay.
And one more thing,
Okay.
microscope. That's it.
Okay.
Flask and all that, they are not needed. So this is just vocab: bacteriocydic, tetracycline, bactericidal, antibiotic resistant. Creating a drug-resistant bacteria. Okay. This is a constant evolutionary race between the pathogen and their host. We keep developing a new medicine and bacteria keep evolving resistant against the drugs. I told you last time that there was a bacteria, means NDLS 25, developed from Central Delhi, and it killed 5 or 6 people in America. And no antibiotic was working against it. Why? Because of the use of the heavy dose antibiotic in India, in Central India, part of the India. So that bacteria become resistant to all antibiotics. So every time you give the antibiotic, bacteria become more resistant. Means next time you have to develop more, you have to develop different antibiotic and more powerful antibiotic. There is a constant evolutionary race between the pathogen and their host. We keep developing new medicines and bacteria keep evolving resistant to these drugs. An antibiotic is easy only effective if microorganism has been binding the site for drugs and metabolic process or biochemical pathway with which the antibiotic interferes. However, a mutation can occur during the bacterial reproduction. Some mutation may help the microorganism resist the effect of antibiotic. For example, a mutation may make the cell wall impermeable to the drugs, right? Now the drug will not go inside the cell wall. As a result, the natural selection advantageous mutation become more common and bacterial population become increasingly resistant to the drugs. Mutation can also result the new bacteriochemical pathway or can switch on the creating a gene for production of antibiotic destroying enzyme. With these mutation, pathogen will become more competitive and evolutionary race. Extensive use of accelerate the processing using the different antibiotic to tackle increasing resistant, increase the selection pressure for the evolution of the bacteria that are resistant to all of them. Evolutionary race is creating what is known as superbugs. For example, methicillin resistant Streptococcus aureus (MRSA). In the absence of effective antibiotic, these resistant bacteria are quite capable of causing death. To prevent this continuing, we must reduce the selection pressure for antibiotic resistance. We can do this in number of ways. By using antibiotic only when they are strictly necessary, means regular or high dose antibiotic must not be used. By making sure that people understand that they must complete each course of antibiotic. By using a few different antibiotic as possible, keeping some reserve for only for everything else fails, right? Some of the antibiotics should be made, which should not be used regularly. They should be used only in those conditions when all antibiotic get failed, right? To reduce the development of antibiotic resistance, we need to vary the antibiotic. Means we need to change the antibiotic every time. We need to uh, uh, vary the antibiotic we use to introduce new antibiotic when possible. The problem here is that we are developing and approving for those fewer and fewer antibiotic. This means the bacteria get repeated exposure to existing antibiotic. Means suppose that this time there is certain skin disease, we are using some antibiotic. We must not keep on using the single antibiotic. We let the other companies to develop more antibiotic for the same disease, right? Different kind of the antibiotic for the same disease, right? By this way, the bacteria will not, because if bacteria is given every time the same antibiotic, it will develop the resistance. But if the bacteria is given different antibiotic every time, it will not develop the resistance for that. This means bacteria get repeated exposure to existing antibiotic, which increases the probability of developing resistance. Hospital acquired infections. Superbugs are commonly found in hospitals and care homes, where the people are ill or have surgery and where the antibiotic uses is at its highest. MRSA, Clostridium difficile, are causing the particular problems. Patients who have become infected with these hospital-acquired infection (HAI) needed to stay in hospital much longer. Based on the recent UK figures, these extended hospital stay combined with the treatment needed to overcome the infection. The cost of average is extra 4,000 to 10,000 per patient. What pound? Some people die as a result of HAI, those hospital infections. A 2012 study, hospitals in the UK found that around 6% of the patients in the English hospital have HAI, including pneumonia and norovirus causing the diarrhea and sickness, as well as MRSA and antibiotic resistant C. difficulty. This is down from 8.2% 2006, but represented the thousands of the patient. Now, what is MRSA? Now, 1/3 of the people, these are the just case studies which are telling the how the bacteria is becoming the resistant. This is 1/3 of the people have bacterium S. aureus on their skin or nasal passages without it causing the problem. If the bacterium enters in the body, it can cause the boils, painful infection, swelling under the skin, or infectious throughout the body, such as septicemia. Many staphylococcus infection have been treated very effectively with uh, uh, methicillin, a penicillin-related antibiotic. As a result, a mutation, some of the bacteria can now produce a penicillin enzyme that breaks down the methicillin. In hospital and care homes, patients who are weak with other infection often develop the opportunity to S. aureus infection and have been treated with the methicillin and with the other antibiotic when methicillin is not worked. The result is that hospitals and the care homes, S. aureus is now winning the evolutionary race because of the almost all the bacterial produced penicillins. Penicillinases, right? What is the penicillinases? That kills the penicillin or break down the penicillin. The infections can treated, but now with a high dose of very small number of the antibiotic. Happening against the control measure reduced the incidences of MRSA from 1.8% patient in 2006 to less than 0.1% in 2012. Right. So that's how we are trying to counter the bacteria who are winning the evolutionary race. Clostridium difficile. What you have to do, Affaf, you have to remember the name of this bacteria and the disease they cause. That's it. Right? Name of the bacteria, please note it down somewhere. Name of the bacteria from the text box, you have to extract name of the bacteria. Next, disease they cause. And next, the antibiotic effective against them. Three things you have to note down in every heading.
Okay.
Right.
Name of bacteria, disease they cause, and the antibiotic that works on them.
Right. Okay. Clostridium, Clostridium difficile. It is an anaerobic bacterium that is found in small numbers in large intestine, about 5% of population. It is not affected by many of the commonly used antibiotic and produce extremely tough spores that can survive in the months outside of the human body of in a healthy person. C difficulty causes no problems because because its number are limited by the competition with the normal gut flora. Unfortunately, some of the common broad spectrum antibiotics are treated to bacterial infections, destroy the normal gut flora at the same time destroying the pathogen they are treating. In some patients, the number of C. difficulty can increase rapidly after the treatment with these antibiotics. These bacteria produces two different toxins that damage the lining of the intestine. This causes the severe diarrhea, can lead to bleeding from the gut and even death. Got it? Sometimes the helpful bacteria become the damaging for body because of antibiotic. So this is a drawback of antibiotic, high dose antibiotic. Infection prevention and control. Codes of practice have been established for the doctor, nurse, and all healthcare worker to try to prevent the spread of healthcare association infection. Means those who are the medical staff, they are getting infected, and to control this effectively. One of my younger brother, he was a doctor, surgical doctor, and after 5 year practice, he left that surgery because one of his friend dies because of the infection in hospital. You won't believe that. This has also happened. So then he joined the administrative service, the medical administrative services. So all around the world, these codes of practice include the following: controlling the use of antibiotic. The careful use of the antibiotic reduces the probability of resistant bacteria evolving. Antibiotic should only be used when absolutely necessary, and every course of the antibiotic should be completed. If the patient takes only a part of course, means taking a small dose, the immune system is unable to cope up with the number of the bacteria remaining and and does not destroy them all. Those bacteria that have some resistance will escape and can infect other people. The use of different antibiotics just encourages the faster evolution of multiple resistance. It is important to reduce the general public who still tend to demand the antibiotic for minor infection and often stop taking the medicine as soon as they feel better. Hospital practice. Good hygiene within the hospital and care homes can have the major impact of the preventing the controlling. HAI example of the good practice include the following: hand washing and hygiene practice. You know what we have to do. Isolation of patient, prevention of infection coming into hospital, right? When the patient are tested, as they must come to hospital, and people who are carrying MRSA, around 3% of the general public are or other infection can be treated immediately and isolated until the bacteria have been destroyed. This reduces the chances of the person becoming clinically ill during the treatment of spreading the bacteria in another, right? So these are the prevention measures. Monitoring level of the hospital acquired infection, right? We need to monitor that. Oh, like what are the number of the hospital acquired infections. Checkpoint. So this was all about the World Health Organization extract. So these are the some factual things, right? Now, Affaf, go through with your this complete chapter and let me know. Go through with complete chapter
Okay.
and tell me if you have any doubt. Then we'll go to the next one.
Sir, are leukocytes and phagocytes different?
Leukocyte? No. Leukocyte is a type of phagocyte. Phagocyte, uh, phagocyte cells are those cells which eat the bacteria.
Okay.
Non-phagocytic, I have told you that is different classification. Phagocyte bacteria, phagocytosis means those bacteria which eat the those WBC which eat the bacteria. Non-phagocytic, who do not eat, they just produces the antibodies. Got it?
Okay. So leukocytes and phagocytes both are a type of white blood cells.
Here, let me make you again clear. Yes, remember this one. Remember this one, Affaf. So I told you, look, lymphocyte. So leukocytes are WBC, right? They may be phagocytic or non-phagocytic, right?
Okay.
So all WBC are called leukocyte, right? Now,
Okay.
first of them, lymphocyte. This is non-phagocytic, means it do not eat bacteria. What it do?
Okay.
It do not eat virus. What it do? Produce antibodies, right?
Okay.
Monocyte engulf bacteria, means this is phagocytic. So leukocyte
Okay.
may be phagocytic or not phagocytic, right?
Okay.
Eosinophil, they are non-phagocytic, means they produces the antibodies. Basophil, non-phagocytic. Neutrophil, phagocytic, means they eat the bacteria. Got it? So leukocyte may be
Yes.
phagocytic or non-phagocytic. Got it? So all those
Okay.
which eat the bacteria or any antigen, we call them phagocytic. Which do not, we call them non-phagocytic. Got it?
Okay. So basophils are phagocytic, right?
Yes, basophils are
Or
phagocytic.
non-phagocytic.
Yes.
Okay. Okay. Understood. Sir, I don't have any other questions.
Another question? Okay.
No other question.
So the PDF we completed, this was the 6B, right? Okay.
Yes.
6B. 6B. Okay. So now the PDF which we have to start, that is a 6C.
Yes.
6C. So we have to start from this. This one, right? Microbiology, immunity, and forensics. So decomposition and forensics. It will be boring, I know.
Oh, yes.
From the heading itself, I came to know this. It must be boring. There should be some clinical test and decomposition. So decomposition, you know what is the decomposition? Yes. Don't you know?
Yeah.
How does they recycle? How does they recycle? How does they recycle?
Good.
Remember or not? Nitrogen cycle we studied.
Yeah.
Ah, mineral cycle we studied. Decomposition in nature. We know that how the bacteria decompose different things.
Yeah.
Remember or not? Yes. We have to
Yes.
read.
Yes.
Okay, that's it. Conditions for recycling nutrient. Yes. When rotting takes place, there should be humidity. Right? Warmer condition that favor the multiplication of bacteria. Like too high or too low temperature, that will reduce the cycle. Availability of oxygen, because most of the decomposition bacteria, they are the aerobic bacteria, right? Aerobic conditions, that should be necessary. In which, okay. Now the next is that investigating of deaths. People die sometimes. They are killed unlawfully or murdered. The murder rate of varies from country to country. For example, the Caribbean island Jamaica, approximately 60 people per 1 lakh population are murdered each year, whereas in Republic of Maldives 2.6 people per billion or per lakhs population are killed in this way. Unsurprisingly, the murder is a crime which has a high profile media. Uh, and it is subjected to many detective stories. To secure the prosecution for the murder, a number of the facts needed to be verified. Who died and who killed them? What was the used to commit the crime? When the victim died, where murder took place, why murder took place, and establish the motive for the crime. Forensic science is an application of science to crime scene. It is important when we need to investigate an unexplained death. It can tell us someone died, what they did, and where a crime was committed, and when the identity of the person who died. Forensic science also gives us the tool to help us and solve the crime, including the murders. We can use the technology for available to identify the criminal or release of the innocent person, but only if the evidence gathered correctly. Investigation time of death. If a body is found, it is not easy to estimate the time of the death. If a person has been died for more than 48 hours, it becomes even more difficult. Forensic evidence is usually combined with the evidence of witness circumstantial evidences to produce the best estimate the time of the death. A number of the change occur in the body any mammal when it has died. It can use these changes with the help of estimate the time of the death. For example, a normal human body temperature is 36 to 37 degrees Celsius, right? It it is kept warm by the energy transferred during the metabolic reaction, such as cellular respiration after the death. Metabolic reaction which have warmed the body being too slow and eventually stop. At the same time, energy is transferred from the surface of the body into the surrounding radiation, conduction, and evaporation of the wood, which cools the body and body reactions. Do not all stop immediately when a mammal dies. So, although the body temperature starts fall immediately after the death, it stay almost stable for the time before decreasing steady room temperature. As a result of the change, the temperature of the body will give some indication how long the person has dead. So, on the calculating the temperature of the body of the person who died, we can tell the approximately time of the death. Number of other factors affect how quickly the body temperature drops. Fat level. The outside of the body cools much more rapidly than the inside amount of the body. Fat affect the rate of the cooling because it act insulation. As a result, a fatter person will cool more slowly after the death than a thin person. That is quite obvious because the adipose tissue they work as insulator. Mode of death. People who died somewhere cold, for example, in a cold water or exposed mountain in winter, will cool more rapidly than the someone who have died in the land and hot climate or heated room. Surrounding the temperature. Our bodies cool faster in cold countries, and if they are placed in freezer, then they do not then do in the hot countries or heated rooms. Surface area volume ratio. The smaller people will cool faster because they have the larger surface area volume ratio than the larger people. Clothing and covering. A body with clothing and covering. A body with the cloths on the insulate it, or the body wrapped in a bed covering or rolled in a carpet will be insulated and will cool more slowly than the body without the covering. Soon after the patient of the death, the body of the mammal starts cool in fairly predictable manner. Despite of all these variations from the analyzing the cool curve of the body known conditions. To ensure the time of the death, you can set in rigor Marriott, right? Okay. Rigor mortis. When someone dies, the heart stop pumping blood. We'll start now. Now we'll do in next class, right? So you have to remember that we have to start from the 147 onward. Okay, Affaf? So read out the last, like go through with the last chapter. And in case if you have any doubt, please note it down. And next class I'll solve those doubt, and we'll start from here on. Okay, Affaf?